Hello, I'm Hanna Dusza with the Collaborative on Health and the Environment. Changejoy is bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's Young EDC Scientist Showcase webinar, or YES webinar, which is titled Potential Endocrine Disrupting Effects of Microplastics in Human Placenta and Aquatic Life. Our moderator today is Sarah Howard, founder and manager of Diabetes and Environment.org. We will leave time following the presentation for a brief Q&A session. You may type in questions to the Q&A feature available on the menu bar at the top of your window at any point during the presentations. After the presentations, our moderator will read out questions for our speakers to respond to. We'll get to as many comments and questions as we can during the Q&A period, and we'll follow up on unanswered questions as we are able. For those who have called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. The webinar is scheduled to last for 60 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Sarah.
Thank you, Hannah. Hello, everyone, and welcome. This webinar, like Hannah mentioned, is a part of a series of started by the mentoring working group of HEEDS, which is Healthy Environment and Endocrine Disruptor Strategies. And we're starting it, or we're working on it as an opportunity for early career scientists to present their research. This is our 6th webinar in the series. If you want to sign up for announcements of future webinars, you can go to heeds.org and sign up for the HEEDS monthly newsletter, and we announce them there. So today our topic is potential endocrine disrupting effects of microplastics. We'll have 2 speakers followed by the question and answer period for both at the end. Our first speaker is Imari Walker, who is a newly minted PhD at Duke University's Pratt School of Engineering. She investigates the release and effects of chemicals from microplastics in aquatic environments. She also works a lot in science communication and has a YouTube channel to share information about plastic pollution, microplastics, and chemicals in consumer products. She's going to talk about her research on how EDCs are released from plastic debris into seabird and fish gastric fluid and the estrogenic effects of these chemicals. Our second speaker today is Hanna Dusza, a researcher at the Department of Population Health Sciences at Utrecht University in the Netherlands. Her research efforts focus on micro and nanoplastics exposure during pregnancy and the consequences of such exposure for fetal and maternal health. She's going to talk about her research on how plastic particles are taken up by placental cells, where they may affect endocrine function. So, Dr. Walker, you may begin.
Awesome. Thank you so much, Sarah and Hannah. All right. So hello, everyone. Again, my name is Imari Walker, and today I'm going to talk about the characterization of endocrine disrupting chemicals released from plastics placed into simulated gastric fluid by utilizing non-targeted analysis along with in vitro bioassays. So before we get into the work itself, we have to talk about why plastic itself. Well, plastic is just a material that, because of its flexibility and its durability, has, become widely demanded since World War II. And its exponential growth over time has led to over 368 million tons produced just in 2019, with the expectation of it to grow. The fact that we can find it in anything, whether that's packaging, consumer products, medical equipment, to even building construction materials, means that we are rapidly accumulating a lot of this waste with the problem of a lack of circular economy to contain it. This means that a lot of plastic that is majority single use tends to end up in our as waste rather quickly, and then has to be remediated in some way. And that means that that waste can also end up in our environment. And that means that it can also transport from land environments to water. Just about 80% of marine debris itself is actually sourced from land. And that includes landfills that blow off debris into our waterways, to water treatment plants that remove plastic debris and filter it out, that can sometimes be reapplied to our agricultural land as biosolids, to even our stormwater, where we're driving our cars and we end up losing tread in our tires, and that tire rubble is still considered plastic debris that can end up in our freshwater systems and then can be transported into our oceans. Just about 1 to 2.4 million tons of plastic actually enter our oceans yearly from freshwater environments. And that can create a new hazard. And once in water, plastic can either float and be transported with the currents or sink into the neighboring sediment environment, where it can interact with all of the environment that it's surrounded by. In fact, the environment that it's placed in can actually affect the process of, fragmentation and release of chemicals. Plastic, once it's interacting with water, sunlight, high temperatures, air, and even physical abrasion processes, can lead to the release of plastic fragments, such as microplastics and nanoplastics, which are highlighted in this webinar itself, and have a lot of media attention and research attention because they can be seen in remote parts of our world, because they can be carried through air and water at such small size fractions, to the fact that not only do these environmental processes impact the release of these fragments, they can also impact the release of these chemicals. All of these processes can increase the release of polymer-associated chemicals, but they can also cause transformation and form new chemical degradation products that could be of concern. In fact, there are many different kinds of polymer-associated chemicals. Roughly about 10,000 are known to be found within polymer databases and can be classified as first monomers, which are the polymer building blocks that are linked to each other to form the polymer chain, like bisphenol A, known to form epoxy and polycarbonate polymers. And then there are this large class of polymer additives and processing aids. These compounds are actually incorporated into the polymer for a specific purpose, such as stabilizing the polymer and preventing degradation while forming that product, to even protecting the polymer during its use. And that's with antioxidants, plasticizers, flame retardants, and even stabilizers. And then there are also chemicals placed within the polymer to actually protect the polymer, to add to the material properties of the polymer, like this dibutyl phthalate, which is used as a plasticizer in plastics. And then finally, there are these class of compounds known as non-intentionally added substances, which are impurities, residuals, and processing degradants that, like in the name itself, they were not intentionally placed. And sometimes the manufacturer does not even know that they're present in these plastics. An example includes semicarbazide, which is a chemical found, which is a thermal breakdown product found. And as these polymer additives and these non-intentionally added substances are not physically bound to the polymer, they can be released into the surrounding water that the polymer inhabits. The release of these chemicals into aquatic environments has the potential to be hazardous. Databases have found more than 98 human health and environmental hazards associated with polymer additives just for plastic packaging itself. One hazard in particular includes being an aquatic toxicant, like 6-PPD. 6-PPD is an antioxidant used in tires. And when we drive our tires and we lose the tread, that tire rubble accumulates on the sides of highways, and during rain events can be washed away into neighboring freshwater environments. That also allows for the release of 6-PPD into water and its transformation into 6-PPD quinone. This chemical in particular is associated with the mass die-off events of coho salmon in the Pacific Northwest. And then other hazards include being an endocrine disruptor, which is the focus of the talk today. But other hazards highlighted include being mutagenic, carcinogenic, persistent, and bioaccumulative. And one of the things that's really important to highlight is that a lot of these chemicals have not been tested thoroughly for their potential to be toxic. In addition to just individual chemicals, mixtures of chemicals can be even more toxic due to the potential for them to act additively, synergistically, or even antagonistically with each other. In a recent study of over 34 plastic consumer products, 74% of these plastic extracts induced a toxic response. Baseline toxicity has also been observed for leachates of common consumer products. And while these studies could detect over hundreds to even thousands of chemicals in both the extracts and the water leachates of these plastics, many could not be fully identified or characterized due to analytical constraints and the lack of reference standards currently in this field for polymer-associated chemicals. And this is important because not only do we need to worry about the release of these chemicals into water, but also the release of them into the stomachs of birds and fish. Fish and birds in particular have potential hazards with plastic debris because they tend to consume quite a bit of plastic. They have the highest count of species itself that consume and ingest plastic debris. And that includes, for types of plastics, those that are commonly found in garbage cleanups, like plastic bags, foodware and beverage containers, and then even things used in the fishing industry. Some of the physical impacts from organisms consuming plastic debris include nutritional dilution, digestive clogging, and starvation. And then the other effects that are really important in a focus of a lot of research these days include looking at lowered energy level, lowered fertility, cell death, and cancer, along with endocrine disruption itself. The motivation for my work in particular was motivated by the study in 2019 by Coffin et al. They took 16 commonly ingested plastic items by birds and fish and placed them within simulated fish and seabird digest conditions. These digest states contained mixtures of polymer-associated chemicals that were released from these plastic items and that were then tested on an in vitro bioassay for estrogenic activity. Of the 16 plastics analyzed, 3 in particular, the polypropylene green string, polystyrene foam, and the polyethylene grocery bag, induced the highest estrogenic activity of the 16 plastics. In addition to just testing the digest for estrogenic response, they also quantified 6 phenols and phthalates within these digests because they are known polymer additives and they are potential endocrine disruptors. And using the concentrations measured, they multiplied them to the estradiol equivalency factors found in literature to get a chemical additive model of estrogenicity. And this result actually just showed that the chemical activity that was quantified did not actually equate to the biological activity in the samples, and that more study was required to better identify some of the chemicals in the digests. A follow-up study of the polyethylene bag, which is one of the 3 with the highest biological estrogenicity, took that sample and chromatographically separated it and fractionated the digest sample prior to doing non-targeted analysis. They were able to identify over 103 compounds via structural annotation, and that included an additional 4 phthalates and 2 alkyl phenols. However, of those chemicals, none were actually confirmed with authentic standards or quantified within the samples to determine how much they contributed to the estrogenic activity in the samples. The objective of this study was to follow up on the previous results and further identify chemicals likely to contribute to the estrogenic activity of the plastics that were submerged in both the simulated fish and bird digest. So in our study, we took 4 of those 16 plastics, the 3 with the highest biological estrogenicity, and then 1 as a negative control to eliminate compounds that were common to the balloon as compounds that were likely not contributing to the estrogenic activity in the other 3 plastics. And those were taken in their digest conditions and structurally annotated with non-targeted analysis along with chemical quantification. And then these samples were also run in the same in vitro assay with the seabird gastric digest of the plastics, the chemical mixtures found of the phenols and phthalates in 2019, and then dose responses of 4 chemicals that were quantified within all of these samples. So let's kind of jump into some of the results real quick. For structural annotation of compounds that were found within the samples, there were over 15 compounds that were structurally annotated using mass spectral libraries within the submerged extracts and digests of these polymers. And then chemical functions of these polymer-associated chemicals included being surfactants, antioxidants, plasticizers, and catalysts. When looking at analyzing these chemicals via models and literature, we were able to find for ToxCast models and test models that there were 8 compounds in particular that were either likely endocrine disruptors or developmental toxicants of some kind. And then based on that information, we chose 4 compounds in particular to confirm with reference standards and quantify in the extracts in the digest for analysis. So of the 4 chemicals, I'll highlight probably 3 today. The first is dichlorobenzene, and this compound is most prevalent in the green polypropylene string for the extracts and the leachates. And this is unsurprising because dichlorobenzene is actually an intermediate for colorants, in particular pigments like pigment yellow 12. When looking at the leachates in the concentration relationship with treatment, we can see that higher concentrations of pepsin and higher temperatures lead to higher release of dichlorobenzene. And we hypothesize this is likely due to the presence of enzymes in the digest conditions that would first reduce the azo bonds on either side of the pigments in the string and lead to the release of the dichlorobenzene with that azo bond reduction. Now, when looking for this, when doing a dose response of this compound in the bioassay, we notice that there is actually a significantly negative estrogenic response. And so this means that it could contribute in some way to the estrogenic response in the green polypropylene string, however, more likely negative. The next compound that was isolated was the tris-2,4-ditert-butyl phosphate. And that is a compound that is actually the oxidized version of a common antioxidant used in polymers like polyethylene. This compound was most significantly quantified in the polyethylene bag extract. However, it showed no significant differences in the concentration by polymer or by treatment in the leachates. When analyzing this compound in the bioassay, it showed a significantly positive estrogenic response in the digest, which is positive. And then a third compound is this Dodecal benzene sulfonic acid. And this compound is most prevalent, again, in the polyethylene bag and has a treatment-dependent response in the leachates where those without pepsin, the not digest conditions actually release far more, and with higher temperatures release more, indicating that perhaps in the digest conditions with the pepsin, that there is maybe some form of transformation occurring to that compound, like oxidation. When looking for this compound in a dose response in the bioassays, there is a trend of a negative response, which has become significant at 20 nanograms per mil. In addition to identifying those new chemicals in the samples, phenols and phthalates that were previously quantified in the seabird digested plastic in 2019 were analyzed in the in vitro assay as chemical mixtures to compare to the additive model that was proposed previously. Now, results demonstrated that the polyethylene bag had the most significant estrogenic response and was comprised of all 6 chemicals, with dibutyl phthalate and diethyl hexyl phthalate being the highest in the mixture. Secondly, the equal mixture of all had lower estrogenic response, but had far less concentrations of both DBP and DEHP. When comparing the estrogenic response in the chemical mixture to the biological response of the seabird digest for each plastic type, we can see that the mixture is actually not significantly different from the response in the seabird digest. In fact, the chemical mixture accounts for up to 20% of the biological activity in the sample. However, when also comparing the biological activity of the digest between now and the 2019 study, we also note that the estrogenic response decreased, and that was also noted in the 2021 Chen study, and could be due to degradation of chemicals within the vial over that time period. Another thing to note is that the mixtures that were meant to chemically represent the polypropylene string and the polystyrene foam were not significant and did not significantly explain the biological activity of the digest, which suggests that those phenols and phthalates likely were not significantly contributing to the estrogenicity in those plastics in particular, which means that compounds like dichlorobenzene and even the tris ditert butyl phenylphosphate may be important to those other polymers. And those two polymers in particular had far higher biological estrogenic response compared to the polyethylene bag and the negative control of the latex balloon. Now, further analysis of the polystyrene foam in particular, because it had the highest estrogenic response, led to chemical analysis of the digest and structural annotation of the sulfur-based compounds that were dominant in the polystyrene foam samples. And may also explain some of the estrogenic activity in the samples. These compounds, however, were unable to be confirmed with chemical reference standards, but were annotated with in silico fragmentation tools. As highlighted here, just the way that things are scored, compounds with the highest score were then chosen as the structural annotation of the compound for these sulfoxides and sulfonyl groups. And so these may be a class of compounds that warrant further study within the polystyrene foam samples and should be further considered for estrogenic activity. Now, just to summarize some of the work here, this study utilized not only non-targeted screening workflows, but also coupled it to light-sensitive bioassays to characterize chemicals and chemical mixtures likely to induce estrogenic toxicity. 15 extractable and leachable compounds were structurally annotated and had multiple functions within the polymer. 4 of those compounds were quantified and demonstrated estrogenic responses. And further, chemical mixtures were significant for the polyethylene bag, but not as much for the string in the foam, indicating other compounds need to be highlighted and identified as well in the vitro bioassay. And the compound class of organosulfur-based compounds definitely requires further investigation for their potential to be endocrine-disrupting chemicals within those samples. And then finally, the further knowledge gaps still remain on the estrogenic responses to multiple chemicals that were structurally annotated in the sample. And those include non-intentionally added substances and environmentally hydrolyzed and oxidized chemicals for which no chemical reference standards could be obtained or tested for toxic effects. And before I end off on this talk, I know I always get a few questions about what can we do to kind of address some of these problems. And the first thing I think about, especially when we're trying to prevent birds and fish from interacting with plastic, we have to prevent it from even ending up in those environments. And so that's usually where I suggest things like coral balls and storm trash traps to capture trash before it flows into our environments. And then recovery systems like these solar powered vessels and the cleaning, the sand vacuums that separate microplastics by density. And these are great and a great step forward. However, they only get a small amount of the plastic that's already in our environment. And most of them can still end up in our landfills and be reapplied as biosludge and continue the same system of releasing back into our environment because of a lack of circular economy. Now, the other option is policy changes and advances. While there are quite a few state and local suggestions for bans and fees, there's also the Break Free from Plastic Act federally that I think addresses more of the question at hand about toxic chemicals. We need to do a more, a greater inventory of the chemicals placed in plastic and their toxic effects themselves. And that also may be important to place in our international treaties that are coming forward in 2022. I know the UN is talking about that Global Plastics Treaty as well. And so with that, I won't hold you all any longer. And if there are questions, I'm happy to chat after this talk, and then you can take down my information if you want to talk after as well.
Thank you so much, Dr. Walker. What an amazing presentation and awesome work that you're doing. While we are waiting for our next speaker, Hanna, to pull up her slides, I would like to remind everyone to put in your questions in the Q&A feature available on the menu bar at the top of your window. We'll begin the Q&A session after Hanna's presentation, and Sarah will lead that. So Hanna, feel free to pull up your slides when you have a moment. Great. It's off to you, Hanna.
Hi, everyone. Thank you, Hanna and Sarah, for the nice introduction and for inviting me here today to speak. So yeah, my presentation is going to be about microplastics and human placenta. And this is work that we have done last year under funding from Dutch government or from Dutch funding body. It was actually a really short project, so I will not go very much in depth about results, but in this presentation, I would like to tell you a little bit more about the importance of researching microplastics during pregnancy in general. So I was really surprised at the beginning when we got this funding, when I started researching this subject in more detail, that actually there has been very little initiatives to look at microplastics in human health. And our research was one of the first one out there. And this was extremely surprising, considering that human beings are probably the most exposed species out there. Really, microplastics has invaded almost every aspect of our daily life. And still, we know so little about microplastic exposure in humans, and specifically in pregnancy or during pregnancy, and what kind of consequences such exposure might have for maternal and fetal health. So my research really focuses on pregnancy for a simple reason that this is the most sensitive period of human development. So any insults that happen during development prenatally can have a long-term health consequences for the developing baby. So because this research is so new, I really would like to focus my talk a little bit as well on why we are studying placenta and why is it important to study it in the context of microplastic research, and of course show you our first preliminary results. So let's talk about placenta. And where it actually comes from. And I think it might surprise a lot of you out there that placenta is not of maternal origin. It actually originates in blastocyst, and blastocyst is a fertilized egg that we see in here. And the cells here in the middle are cells that will give rise to a baby or to an embryo. And the cells that surround the blastocyst are actually the first placenta cells. So this little blastocyst at around 2 weeks of pregnancy will implant itself into the uterus of the mother. And over there, these first placenta cells will start to proliferate and divide quite rapidly. And they start to dig in through the uterus wall in order to get to maternal blood supply. And the only goal for the placenta right now is to really suck up as many nutrients from the mother as possible in order to support the developing baby. So the placenta cells are growing and proliferating and digging deeper. And at the third trimester, the placenta becomes this amazingly complex organ. It's the size of a volleyball right now, and as much as 25% of maternal blood can actually flow through the placenta. So you can already imagine that if there is anything present in maternal blood, it will eventually come in contact with the placenta as well. So placenta is this beautiful, amazing temporary organ that not only provides oxygen and nutrients to the baby, but it also removes waste products. And it is a really important temporary endocrine organ that produces a lot of different hormones that are very important or crucial for maintenance of pregnancy, but also on fetal, crucial for fetal development and maternal health during pregnancy. So why are we concerned with microplastic and placenta? Is there any evidence out there that this might be a cause for concern? Actually, as I mentioned at the beginning, there are actually no data so far, or very little data on exposure to microplastics during pregnancy. And we don't know yet what kind of consequences they may have. But we can speculate based on the air pollution particle research. So air pollution particles are particles that come from combustion, from smoke, from dirt and dust, and they are generally referred to as a particulate matter. And in 2019, so also quite recent, our colleagues from Belgium were the first one to find some of those particulate matter particles, specifically black carbon particles, in the placenta samples, in the real human placentas. It was the first proof of principle that the air pollution particles can actually reach the placenta. And since then, a lot of epidemiological studies shown association between exposure to air pollution particles and adverse pregnancy outcomes, such as hypertension disorders, premature delivery, miscarriages, and even stillbirths. So of course, carbon particles are not microplastics, but size-wise and maybe morphologically as well, they can be quite similar to microplastics. Moreover, what we actually see is that microplastics are becoming a big source of air pollution. And this is a report that came out in 2017. It talks about primary microplastic sources in the ocean. So what happens is that a lot of microplastics are released in the air that we breathe as well. They can travel along huge distances, and then together with the rainfall, they can fall into the ocean. And the sources that they found that were the biggest sources of microplastic pollution were actually synthetic textile, car tires, and city dust. And more research recognize that microplastic is this new emerging source of particulate air pollution. So, of course, this is not the only source of human exposure. We are also exposed with the food that we eat on a daily basis, and water that we drink on a daily basis. Microplastics are literally everywhere, and there are multiple exposure routes. So in our research, we really wanted to ask questions. Can microplastics be taken up and transported through the placenta? So looking at the black carbon particles, is that similar? Can microplastic cause harm to the placenta? And can the chemicals associated with microplastics also cause harm? So how do we go about and actually study this? We can grow placenta cells in the laboratory, and we use so-called vivo B30 cells. We can differentiate them into two distinct cell types, non-syncytialized vivo and syncytialized vivo. And these two cell types are representative or very important placenta cells called cytotropoblasts and syncytiotrophoblasts. So these two cells are actually in direct contact with maternal blood within the placenta, and this is where all the exchange of nutrients and oxygen and waste product takes place. Moreover, these cells are also extremely important for metabolizing or producing a lot of different hormones that are crucial for pregnancy outcomes, healthy pregnancy outcomes. So extremely important cells to investigate. And what we have done in this very short project, actually it was only a half a year, we took that 2 cell types and we tested common plastic particles such as polystyrene and high density polyethylene particles that you probably already heard about. And we also weathered these particles in experimental setup in the laboratory to mimic environmental weathering. So we wanted to see if the weathering changes the chemical composition of the plastics and has any impact on the uptake and eventual toxicity of these particles. So we tested the pristine and weathered particles in the 2 different cell types, and we looked at different endpoints such as the uptake, cell viability, damage to plasma membrane, and gene expression. We also look at the transcellular translocation of these particles. So can they actually go through the placenta cells into a deeper layer of the placenta? And at the end, we also chemically characterize the pristine and weathered particles to see what chemical changes the weathering may induce. So let's look at the uptake. What we see in here is placenta cells grow in monolayer, and we visualize uptake using confocal microscopy. We can actually stain different cellular compartments. So the pink color in here, it's actin. And actin, it's a protein that is present within the cell. It's some kind of a cellular cytoskeleton. And this is visualized in pink, sorry, in here. You can see that this dirty yellow colored are actually the nucleuses of the cell, and the little fluorescent particles are the 200 nanometer polystyrene particles. And let me play this to you. So what you can see is that these particles are really internalized and they are taken up by the placenta cells. And we have done this with other particle sizes from 0.2 micrometers to 10 micrometers, and we saw that all of those sizes have been taken up by the placenta cells. Even the 10 micrometer, which is actually really a huge particle, almost a half of the size of the cell itself. And this uptake has been observed in both non-sensitized and sensitized cells. So then we were wondering, if we see uptake, can we also see the transports? And we look at the transport by using so-called transwell setup. And we took the 50 nanometer, 200 nanometer, and 1 micrometer polystyrene beads. And what we notice is that all of those particles were also transported through these placenta cells. What is important to note is that the transport was size dependent. So the 50 nanometer particles transported through very quickly through the placenta cells, whereas the 200 nanometers and 1 micrometer were much more retained within the cell. And what that tells us is that when there are plastic present in maternal blood, not only they can accumulate in the placenta cells, but smaller particles can also travel through and possibly expose the developing baby. So we know there is an uptake, we know there is a transport. Is there also a toxicity? And as Imari already mentioned, we cannot talk about toxicity without talking about all those different compounds that are also leaching out of plastic particles. And she also mentioned that there are more than 10,000 different chemicals that are associated with plastics. And these are colorants and fillers, flame retardants, plasticizers, and the list go on and on. So this is really important to remember that there are plenty of different chemicals that can leach out and might cause toxicity, not only particle toxicity we're looking at. So we first took the pristine and weathered particles, and we wanted to take a look at the simple cell toxicity assays such as cell viability and damage to the plasma membrane. And what we notice is that none of our particles have actually caused cytotoxicity or acute toxicity of different size ranges and a different concentration range. And in here we tested concentration up to 100 micrograms per milliliter, which is really, really high. And of course, in a real life scenario, the exposure to these particles might be much longer in a real placenta tissue. So we cannot exclude the cytotoxicity can be present in the chronic exposure. But nevertheless, in our research, even 100 micrograms per milliliter did not cause acute toxicity. So then we wanted to also look at the gene expression, and we were very much interested in genes that are responsible for producing different hormones and are involved in steroidogenesis, so basically producing steroid hormones during pregnancy. And we investigated 5 different genes and exposed ourselves to pristine and weathered particles. And what we actually saw that the pristine particles show effects on gene expression, or 2 genes, whereas these effects were much less pronounced in the weathered particles. And these 2 genes are genes that are extremely important from production of 2 hormones, estradiol and testosterone. And both of these hormones are crucial during pregnancy. Not only they maintain pregnancy, but also they are involved in fetal brain development and sexual maturation, differentiation, sorry. So really, really important endpoint. This is just the preliminary data we investigated very recently. So we have to look into this in more detail, but definitely an important endpoint to keep an eye on. So what we wanted to take a look after is actually if there are any chemical differences between the pristine and weathered particles. And we performed the non-targeted high-resolution LC-MS analysis. And what we found is that actually pristine particles had many more unique chemical features than the weathered particles. And what that tells us is that the weathering process can change the chemical composition of the particles. Another interesting thing to note is that we performed the partial least square analysis, and which basically tells us that there is a difference between different samples and where that difference comes from. And what we saw is that the weather and pristine particles formed separate clusters. So as mentioned before, this is a difference in chemical composition between the 2 conditions, weathered and pristine. But also what we saw is that for the pristine particles, there was a clear difference in chemical composition mediated by particle size. And that was very, very interesting indeed. So this basically gives us a first indication that weathering and size might be important determining factors in the chemical composition and toxicity of microplastic, and this has to be considered in the future investigations. So we are still investigating which which chemicals actually could be responsible for the effects observed in here. So I can't give you that information right now. And this is just a short summary of that project. And I think we can just summarize, which is very important, that there is uptake, there is transport, and possible subtle toxicity on endocrine functions of these particles. And considering the widespread human exposure to microplastics, this is definitely a cause for concern that needs further investigations. And I cannot continue this without mentioning 2 publications that came out just very recently. And this is really hot from the press. This is research that came out a few months ago, I believe. And this is the first research that show microplastics in the real placenta samples as well as fetal feces. So not only it shows that the particles are actually getting to the placenta, but also that it's transported through. So emphasizing also the research that we've done and supporting the research that we've done. So there is no longer a question if those particles reach in your environment, but we still don't know to which extent these particles are present in utero. And of course, we need to know to which extent they are present if we wanted to talk about any risk associated with them. So fortunately, the problem of microplastics and human health has been recognized nationally and internationally. And there are new research initiatives founded just this year that will investigate this further, both on the exposure assessment side as well as the hazard identification side. So we have the Momentum project founded by ZonMV, which is a Dutch funding body that would look more broadly on human exposure and passage of microplastics through different barriers, not only placenta, but also lung and gut and brain-blood barrier, for example. And we also have Aurora project, and this project was funded by European Commission, and it will focus very much on the exposure during pregnancy and early life health. And both of these projects will use cohorts and real human tissues to investigate what is the extent of internal exposure to microplastics in humans. So in here, not only we learn more about this issue, but also we wanted to provide a long-term roadmaps and solution routes to minimize the health impacts of microplastics. And these solution routes can be various, from advancing analytical methods to actually be able to measure those microplastics in human samples, as this is a big gap in the research right now. Look at policies to reduce health impacts of micro and nanoplastics, perform large-scale human health evaluations, and use new products that are safe by design. These are all just some of the solution sets that we would be working towards in these two projects. But of course, we should not forget that microplastics are not the only environmental stresses in the uterus. And I wanted to focus, as this is an endocrine disrupting forum, a little bit about, I wanted to talk to you a little bit about my PhD, where we performed the research and developed methods to look at endocrine disrupting compounds in amniotic fluids, in human amniotic fluids. And what we found, which is not surprising anymore, is that there were plenty of known EDCs as well as novel unknown compounds present in amniotic fluids. And some of these novel compounds were potent plasticizers like diphenyl isophthalate, for which we found very limited to no information on production and use. So this research on microplastics and EDCs really shows that there is a need to better understand the source, nature, and extent of exposure to these multiple environmental stressors during pregnancy. And I think combining these multiple stressors in risk assessment is going to be very challenging, but it's a necessary step forward in order to fully protect the next generations. So I think I would like to leave you with that. But first, I would like to thank people that were involved in this project, specifically Juliette Legler, which is a continuous source of inspiration and knowledge. And I would like to thank our funding bodies, ZonMV and Health Holland, for recognizing that this is an extremely important research and for supporting us. And of course, I would like to thank you all for coming here today and listening to our talks. And I hope that after my talk, you can have a little bit bigger appreciation for the placenta. Thank you very much.
Thank you. That was great. Both of the speakers. Thank you so much. All right, now we are going to open it up to question and answer period. And I will read, if you can type your questions into the Q&A box. I will read them off. And I'm going to start going backwards to Imari. I know this one was for Imari. It says, what in vitro method was used for the digests?
Yeah, so this is I'll give you the full name, the VM7 loose 4E2 cells that were combined with an estrogen receptor for ER alpha, ER beta, responsive firefly luciferase reporter gene assay. So they were measuring luciferase using luminometer.
I was going to say got it, but I'm not sure. I don't really got it. And then this is also related to that. How many of the chemicals detected in the digest may accumulate in the fish and thus may be a source of human exposure?
That would be a tricky one. I mean, I do have some log KOW values that might give a little bit of indication, but gosh, I mean, at least half of those that were structurally annotated were high enough, I think, to at least end up in the octanol portion, and that may eventually be bioaccumulative, but I would have to do some pretty thorough research on every chemical to have that information.
Thank you. Now I'll switch to Hanna. I keep saying the American.
No.
How similar are the microplastic exposures in vitro to what we're exposed to in the environment?
Yes, this is exactly what we are struggling with. They're not that similar, and it's extremely difficult still to extract microplastics and nanoplastics from the environment in quantities that we can use for toxicological studies. It's although there are so many of them out there, that the technology is just not there yet. So we use the particles that are well defined and characterized, also to minimize the variability that we are looking at. And this was the first step, but I can promise you that in our new research in the Aurora and Momentum project, we are actually trying to do that, to move from those well-defined particles into more human exposure relevant particles, and we are making them ourselves and environmentally weathering ourselves and trying to mimic the real human exposure as close as possible. So hopefully we can have data on that soon. But it's a very good question, and yeah, critically looking at these particles, it's important.
Mari, do you want to address that too, or?
Yeah, so mine's a little different. They're not necessarily microplastics for this chapter of my dissertation, but they're pieces of plastic that you commonly find on the beach that were cut into sizes small enough for a bird to consume. So technically in the environment, you probably would see a whole bag, which, I mean, maybe the bird or fish could consume that whole bag on accident. But yeah, they are pristine in their original form before they undergo that digestion. So environmental relevance, I mean, a brand new bottle thrown on the beach, if something encounters it, still possible to find those effects. No different than if you just, threw a styrofoam cup onto the beach. I mean, those can be also quickly targeted and consumed in some way. But yeah, that's a difficult one to target the environmental relevance with weathered plastic.
Yeah. Okay, great. Okay, Hanna again, nice talk. I mean, both have nice talks. I wonder, why did you measure the expression of CYP11A1 in placenta cells? Does the synthesis of 11-hydroxyandrogens occur in placenta cells?
Yes, it does. So I was quite puzzled at the beginning. Of course, there are plenty of different genes that you could look at. And what I've done is actually I went to literature and just searched for the ones that we already know that plasticizers might affect. And I knew that this gene is as well expressed within these placenta cell types. So assuming that there are a lot of plasticizers in the plastics that we are using, I was curious if I can see any effects of those plasticizers leaching out maybe of particles on these genes that were already affected by other plasticizers in the literature. So this was basically a little bit how we approach this. But of course, you know, these are just few genes, and it's not a full selection. So we needed to start somewhere, and this is where we started.
Can also, and then someone asks, why do you think the size of the pristine, you know, particles affects the effects that they have?
Yeah, I think this is largely to do with surface area, maybe. And this was very new, something very new that we saw, but I think it has to do with the surface area and how those particles can leach out from, how these chemicals can leach out from particles if they have different surface areas. So there are a lot of different chemical how do you say, activities happening on the surface of the particle. So it's probably related to that. And of course, smaller particles have bigger surface area. So more leaching of chemicals can happen with the smaller particles. So yeah, this would be my first thoughts, but this is not something that we have investigated in much depth yet, and I don't think anybody did. So this is, as I said, very new, and we still need to look into that.
Imari, do you have something to add to that too?
No, I was going to say something similar, just that there is a size dependency with release of chemicals. At least that's what I've seen in some of my water leachates and previous work, that, you know, nanoplastics, microplastics tend to leach far more toxic chemicals. And I think there is a study for polystyrene nanoplastics in particular that shows smaller size releases more toxic chemicals.
Interesting. Imari, someone thanks you for raising the solution of producer responsibility in your talk. And quick question for Hanna. It seems like the reanalysis of biobank archived placentae could provide us with some long-term outcome epidemiology on placental microplastics. Do you know of any work done in that area?
No, but we do have biobanks that we are using as well, and some samples that has been stored for different cohorts back in the days that we will use in Aurora project. So there's a lot of placentas in the hospitals waiting for us to analyze. Very exciting about that. But I would really be interested into looking at the historical placentas, if you might call them like that. So placentas that were maybe stored before 1950s, before the plastic production really became so widespread, and that would be very interesting, maybe to also see you know, the difference in expression of some genes with placentas from 1950s or before, and the new placentas, and different biological markers of cellular stress, oxidative stress, immune toxicity of particles, and so on. So yeah, there's plenty to do, and this is a very, very good point. So we will definitely look into that more in our Aurora project as well.
And then someone asked, probably, Imari, this might be, is there a particle size limit that determines a microplastic if it can be absorbed or leach versus not being able to be absorbed? I mean, it seems like the wildlife is just eating full plastic bags, you know. And I don't know.
I guess it just depends on your endpoint. If we're talking about absorption through the skin, there's limitations into size limits into, you know, the skin barrier. Ingestion is just how much can they actually eat on the esophagus, you know, barrier. but we're, none of those, at least when we're talking about ingestion, you can easily eat pretty large pieces of plastic. And we define sometimes microplastics being up to 5 millimeters in size, but that's changed widely throughout research, what we're going to set the cutoff to. But most of those would be easily ingested by quite a variety of different organisms, especially birds and fish.
She's skin barrier, please, if we know.
Skin barrier is more likely nanoplastics. I mean, I think a lot about glitter. I think a lot about eyeshadow. I think a lot about the fact that we just apply so many different things to our skin that we probably haven't studied widely enough on that nanoplastic, microplastic range that may end up being absorbed into the blood and may end up transporting to a placenta. I don't have answers for that, but I think that those are entirely possible.
All right, well, thank you. I think we have to be done now. We're out of time, but thank you for your talks, and it was really interesting.
Thank you. This is wonderful.
Thank you.
Great. Thank you so much, Dr. Walker, Dr. Dusza. I wanted to say a big thank you to you and to Sarah for moderating today. We're approaching the end of today's webinar. A video recording will be available on Che's website soon, and tomorrow you'll receive an email containing a link to the video. The next Che Alaska Partnership webinar will take place on December 7th and is titled From Exploration to Closure of the Large-Scale Mine, Environmental and Health Impacts of a Proposed Donlin Gold Mine on the Watershed, Communities, and People of the Yukon-Kuskokwim region. Then on December 15th, the next Che EDC Strategies Partnership webinar will host a webinar on PFAS exposure and epigenetics in the firefighter cancer cohort study. To learn more into RSVP, please visit our website at healthandenvironment.org. If you are new to Che and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on your website at healthandenvironment.org. Additionally, if you appreciate these Che partnership webinars bringing you the latest environmental health research for free, we encourage you to support Che's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Imari and Hanna, for taking time to present today, and to you, Sarah, for your excellent moderation. Thank you so much for joining us. We're wishing all much health and wellness. Stay well. Take care.